Okay, recording's on. Before we get started, I'm going to go ahead and share my screen. Are there any questions from any of the content that we went over this morning or yesterday? Is there anything that anyone wants to see again? Okay. So before we carry on with our FastQ Accounts Jupyter Notebook, I want to make sure that all of you guys are still logged in to the SMCE system. So everyone go ahead and check their browser to make sure that they're still logged in here. Also, please check the lower left-hand corner of your screen to make sure it says Python Conda environment GL4U, and it should say idle. So please make sure that's there. If you guys are seeing anything other than this, I want you to go to the upper right corner where it tells you the kernel that you're in and click on it. And then in the drop-down menu, you're looking for Python Conda ENV:GL4U. You want to click on that and click select. And then just make sure down here, it should say idle, Python Conda environment idle. And then if you did have to restart your kernel, remember anytime you restart your kernel, you lose all your variables. So if you did have to do that, I just need you to come up to this section, the define paths, and just rerun that code block to reset your variables. Okay, so I guess I haven't heard it from anyone yet, so you guys are all good? You all see the Python Conda environment GL4U idle? Just before lunch, we took a look at the Slurm script that you all submitted for processing FastQC, so for running all of our raw FastQ files, which contain all of our raw reads, through the FastQC tool, which will assess the quality of the reads in the raw FastQ files for each samples. And so you guys all submitted that through this sbatch command here. You got a job ID back. We went through this script. I know this is kind of a little over really the what we do in the course. A little bit more scripting in here is involved, but I did just want you guys to know how to run multiple jobs through a Slurm array. So if you guys have any questions on the script, I'm happy to answer them. We're not going to look at the script for every single tool we run unless you guys really want to. It's all set up very similar. So what we are going to do though is definitely look at the actual command that's run for every single step. But if you guys do want to view the scripts, they're all here on the system, so we can view them if you guys have questions or want to see them. So that said, let's go ahead and go down to this code block here that starts with cat, and then you see it should end in .out. And so I should point out quickly from the script. So as I mentioned, this processing is in variable that we had set up for a path to where I put all the processing scripts that we're going to be running. And then I have a subdirectory set up within that main for each level of the data that we're going to be processing. So all the processing scripts for raw data are in a subdirectory in that path called 00 raw data. And then I have another subdirectory here for all of the standard outputs that are run for each sample. And then what you'll see in there is a standard output file for each sample that's run. Notice the name of this file. It comes from the script that we ran. So as a reminder, this is the Slurm script that we ran. And we told Slurm that we want to put all of the standard outputs in a subdirectory called raw FastQ outlaws logs. And we want to name each file glds104_raw_FastQC. This %A is going to be equivalent to the job ID that you're given, well, that I was given. So it's going to be equivalent to the job ID when you submit your batch job, right? So if you're all run this on a real job scheduler, your batch ID would be, mine in this case would be 440, yours is going to be different. And then we had this underscore percent little a, and that's going to represent each job in the job array that we did. So again, associated with each of the 12 samples, and this is just going to be a number 1 through 12 corresponding to the sample that was run in that samples.txt file. And so that's why when we want to look at our standard output here, it is indeed in a subdirectory called raw fastq outlaws, and then the name glds104 underscore raw underscore fastqc. This was the job ID that I was given when I pre-processed these data, and then you see underscore 1, and that's going to correspond to the first sample that was in the samples.txt file list that I provided in this part of the script. And that first line of it is going to be the first sample, which is our first replication, first biological rep of our flight samples. So we can go ahead and run that. And you should see something that looks like this. Is there anyone that's not seeing that? Okay, so I assume you guys are all looking at the same thing I am on my screen. And so just some of the stuff up here is some of the stuff that I kind of added to the script here. So just to kind of look at this side by side to see where this information came from. So this is the stuff here, right? Remember that in the script, I had this echo command to echo glds underscore raw underscore fastqc. So that's why in the output, we're seeing glds 104 raw fastqc. I added a space here, so that's why there's a space before you see the next part. I then ran some commands to indicate the start time and then to print out the start time in seconds, which is here. I told it to print out the server name that was ultimately used to run this job. And so the name of the server where this computation was done is called DPGPU1. I then asked it to spit out the Slurm array ID. So this should be number 1. We know that from our output. So that small %A that we have in the script here should indicate the number of the array job that it is. And so we see a 1 here, which means that this should be Slurm task ID number 1. And then I also told it to activate a certain environment, so there's no standard output associated with that. I defined variables, but there's no associated output with that, as we know, unless you take a look at what your variables are. And then I asked it to define my samples. And so again, assigning a variable, and then I told it to print a space. That's what we're seeing here. And then I told it to echo sample followed by the variable held in that sample variable here. And it should be the first sample listed in the samples.txt file, which is flight rep 1. So that's why that's there. The other thing that I kind of add in here is to print out the version of fastqc. So that's why we see this here. It is version 0.11.9 of the fastqc tool. And then in the script, I tell it to actually, I have my command, my fastqc command stored in a variable, and I tell it to echo that variable or to print out that variable. So it should print out my command. And so you see here the exact command printed out. The nice thing about this is it'll add in everything that's kind of stored in a variable here, right? So we have my out directory stored in a variable, the sample name stored in a variable. But when I print it all out, it should paste what those variables actually are. And so that's why we see here that the out is the actual output directory, which we all set up on our system. And then the input file. So, and you saw that we had 2 input files because we used in the command that wildcard there. So it should be the sample name, which is our flight rep 1. It should be the R1 file and also the R2 file, because that wildcard indicates that could be anything so long as it ends in underscore raw fastq.gz. And so that's what we are seeing in our standard output here in the command. So it'll first run R1, and then we see that it also ran fastqc on R2. And then the standard output for fastqc is it essentially prints out a log as it runs through and looks at all of the reads and does the evaluation of all the reads. So it'll basically take it and break it down into percentages, and then it kind of spits out in your standard output, okay, 5% of this is done, 10%, 50%. So every 5% it gives you an update. and it tells you that it's working on the forward read first. That's the file that it's acting on. And then you should see that the analysis is complete. That'll tell you that, okay, it successfully completed the analysis. We don't see any errors. So if you remember when we were running Unix, anytime we had something wrong with our code or when there was an issue, if you guys remember when we did that and we saw errors printed out. So this is what you're looking for when you're reviewing the standard output of these tool scripts, is you want to make sure that you're not seeing any errors or anything that would be of concern in the standard out. And for FastQC, you want to see that it says that the analysis is complete for the file that it's working on. And then remember that we had that wildcard there, so it should have done the forward and the reverse read. So you see that right after it completed the FastQC analysis for the forward read, it then started the analysis for the reverse read for that same sample, flight rep 1. And then again, it gives you kind of a log just to let you know every 5% that it's complete, that it's completed. And then again, you should see that it said that it's complete for the reverse read of that flight rep 1 sample. And then if you remember from the script that I have kind of this series of commands here that basically just calculate the total time that the job took, and I calculate it in seconds, minutes, and hours. So you notice that when I processed these two samples, it took about 20 minutes, so roughly 10 minutes each, which is why I didn't want us to have to sit here and wait for everyone's to complete. And so now that's how we're going to know, looking at the standard output of our FastQC analysis, that it completed the FastQC analysis for our forward read, and it also completed the analysis for our reverse read. And Elba, I think that you just joined, so just make sure that your kernel's still active, and it says Python conn environment GL4U. If not, then you can go here and select that kernel, click select. If you do have to restart your kernel, then you'll have to rerun this code block here to redefine all of the variables that indicate all the paths to where the data are that we're looking at.
Thank you. It's still running, I think. I'm not as far with you. I got bumped out of web, but not off Jupyter Notebook, so I don't know how that was about. But anyway, keep going. I'll catch up.
Sure. So I think all that you missed is we got back and we just took a look at our standard output. So we're at this part, the cat processing the file that ends in fastqc.out. And so just took a look at what the standard output for fastqc looks like. And just to summarize it, basically we should see a log of every 5% showing that the fastqc is running for this first file, which is the forward read of our first sample, which is flight rep 1, followed by the analysis for our reverse read for flight rep run. And again, the program is set to give you a kind of status output every 5%, and then at the end it tells you that the analysis is complete. So what we're looking for, if we've set up the command to run both the forward and the reverse read in the same command, which is what we did here because of that wildcard, we should see 2 analysis complete, one for the forward and one for the reverse read. And that's how we know that we've successfully completed this job. And then just a note that it took 20 minutes on the system that I was working on to run that. And so the standard outputs that we have, so this are the outputs that are generated from the FastQC. So this is a standard output, which is usually just what the tool gives you as kind of a status. Any errors are going to be in your standard output, but all of these tools are going to generate some output files. So if you scroll up a little bit, you'll see when I wrote the definitions and the input and output data, that this tool will generate 2 output files. So it's going to generate an HTML file, which is going to be the FastQC report. That's what we went over in the lecture. It's also going to output a compressed folder, a zip folder, and that folder is going to contain all of the data that's used to generate the graphs and the nice visualizations that we see in the HTML file. So those are the outputs that we should see, and we directed those outputs to go into this folder. So we directed the outputs with the -out flag to go into our nice directory structure that we set up here. And so again, we kind of simulated this environment so you won't actually have those outputs in there, but we'll show the outputs that you would get. And so that is going to be in an HTML file that you copied to your home directory. So if you remember, after I had you guys set up your directory structure and then create the variables to the paths to where all the data are that we're going to be viewing, we then ran some copy commands to copy HTML files to our home directory because for whatever reason, Jupyter has a hard time showing files that are within the file system. So I had to copy them into your home directory. And we're running a Python kernel right now, right? And so in order to view HTML files, we need to load a library from Python in order to view HTML files within this Jupyter Notebook in Python. So I need you guys to run this code block here that is from python.display import iframe. And that again is what we need to activate in Python to allow us to view HTML files. So go ahead and run that code block. Make sure that it completes here. It should be populated with a number in brackets. And then you want to go ahead and run this command. So this is using an iframe command from Python. This iframe command from Python is telling it to open up this file. In this file, notice how there's no path information before it. So it's going to look for this file in our current directory. And we all should still be in our home directory as our current directory. And that's one of the files that we copied over. And then we could tell it to set some parameters for how we want to display the HTML file. And so when I did it on my screen, a 1500 width and a height of 2000 worked well for me. If it looks weird on your screen, you guys can play with the width and the height and rerun this. So when you do run it, what it's going to do is it's going to open up that HTML report of the FastQC analysis that we just did on our first flight rep 1 sample, the forward read. And so if we take a look at that FastQC report, if you guys remember from the lecture, I kind of copy and pasted some of the panels that we see here, but this is what the actual FastQC report looks like. You guys should, when your cursor is within the report, you should be able to scroll up and down to see things. If it doesn't show you all the way to the end, you also should be able to scroll left to right. And again, you could set the window that we're viewing here to display this information is set in this command up here with that width and that height value. So if you want to make it show things a little bit longer than what's displayed here, you can increase your height. If you want to not have to scroll back and forth, you could increase the width of the display. But for visualization purposes, I think hopefully you guys can all see my screen here. And so I'm going to go over this report. So you can click to any part of the report. This is an interactive file. And so if you want to skip, like if you're only interested in seeing your per base sequence quality, you could skip around in it. So we're just going to take a look at this file, and then I'm going to prompt you guys to ask I'm going to ask you guys some questions about the information here. So again, this should look familiar from the lecture. So we're seeing basic statistic information. So you know this is where you find things like total sequences, sequence length, %GC summary. And then if you scroll down, I went into great detail in this in the lecture. So if you guys have questions, we could talk about it, but I'm going to have you look at this and answer some questions. This is the per base quality that we went over. So again, base pairs from left to right, the position on the left-hand side here is going to be that Q score. So that should look familiar. This other one that we haven't gone over, this is a per tile sequence quality. And honestly, this is showing you per tile, so it's looking at specific segments of where these reads would be on the cluster of the flow cell. I'm not entirely sure how to interpret this. I don't usually pay too much attention to it. If anyone, I don't know if Lauren and Mike, if you guys have looked into this since we talked about it, but feel free to chime in if there's anything you want to say about the per.
I'm getting a file not found. Should I go back and run everything again?
Yeah, well, not everything. So if you're getting a file not found, the first thing I want you to do is add a code block and run pwd
Okay,
and make sure that it's in home Jovian.
okay, hold on. Yes, I am.
Okay. So you might not have run a copy command. So if you scroll all the way up to the top here
Mhm.
and go to the define, no, not define paths, just
Let
under
me do all
the
those, yeah.
define path section to this copy command.
Let me do all of those. Okay. And I'll just do it again.
Yeah,
Okay.
and if you just run this block here, the
Uh-huh.
CP commands, this is copying those HTML files to your home directory.
Okay.
So once you run this, you should see the HTML file. The one that we're looking at now is flight ref1 r1 raw fastqc.html. So just make
Yeah.
sure that you see that on your left-hand panel. And then once you see that in your left-hand panel, then you should be able to jump back to where we are and
Oh,
run this block first to import the library to view the HTML file. And then you should be able to run this iframe command and see it.
can't, how do we get back to that root folder? I click on the folder and it doesn't seem to get me there. Okay, I did it now. Okay.
Awesome.
So I see the reports are there.
Good. Did you run this code block, the import libraries for HTML visualization?
Yeah, yeah. I'll just go ahead and keep going. I'll catch up somehow.
But share your screen.
I
It's okay.
like puzzles. I like puzzles.
It's okay. You can share your screen.
That's okay. Let's go ahead and check I did everything. I had that interruption. Here's the fast queue. I'm just going back and running everything. There's a slurm. Let me try again. Okay, here we go. Still not found.
You want to share your screen?
Sure. Let me find out where it is. Here are all of these.
Yep. So you
You
can
can
go
see here
ahead.
on the left. You can see what's there on the left.
Yeah. So it does look like it's there. And then when you added a code block and ran PWD,
Yeah, I did that down there. It says I'm in home. I mean, I can do it again. I'll do one here. And
okay,
there,
that's
in
good.
Jafian.
Yep. Scroll
We looked
down.
at the slurm. I don't know if, would it not have, and that's just looking at the output. Should I do this again?
Nope, you don't have to do that again. You can scroll down. So
It
run
says this
one,
import.
import, run that one again.
I run that one.
And then,
Go here and I run this one. And it says file not found.
Lauren, do you have any idea why she wouldn't? I can't see an
I
obvious
I
reason.
cannot. Yeah, I'm stumped. You have the file?
And here it is again. Run the next cell. Let me try it again.
Huh. Why don't you try restarting your hub maybe? I don't know why it
Just
wouldn't.
log out.
Yeah, or you could just like refresh your browser.
Reload.
And then just,
The HTML files are still there.
yeah, they should all be still there. So if you scroll down in your notebook and just rerun the alias or the variable setup, that one a little bit higher,
This one here,
a little bit higher.
52,
No,
cell 52.
no, up higher. That
This
one right
one,
there. Yeah, rerun that.
27.
Yeah. Okay. And then you can scroll down. Rerun your copy one just in case, cell 52 that you have there.
An asterisk there, 59. It popped up, so it seems like it's okay. Why is it out of order? Why is it because it's the 59th command I'm doing? Because it's not
Yeah.
in order.
Yeah, it's 59th block that you ran. It really should be like 1 or 2. That's weird that it didn't restart the kernel when you refreshed.
Yeah, it's been strange today, I thought.
And
For
then just,
example, I logged out, and when I went to the page, I was still in. It didn't really log out. That was very strange.
yeah, let's see if this works. If it doesn't work, then we'll like go through and stop your server. And
Okay.
sorry, I think it's important to go through this just because when you guys are teaching this at your home institutions, just in case you or your students experience this, you can scroll down and just run the,
Going.
yeah, so run that 41
Should I
again.
do the display again?
Yep,
Okay.
yep, and then run.
How do I know that how do I confirm that iframe is there?
So it should populate in your, I'm pointing to the screen like you could see, where it says 60, right? So if you were to run it again, that's going to populate probably to 61. So if you go in that cell and hit play,
Here?
yeah, and then run that. So you
Also,
Yeah,
if you don't see
61.
an error,
yeah,
if you don't see an error, then you know it worked.
Okay, here we go. Nope, it's not working. So I think we need to restart the server.
yeah.
Something's
So
wrong.
go ahead and go to File. You don't have to
Mm-hmm.
exit that. Just File,
Log out.
Hub Control.
Mm-hmm.
Click on that. Go to Stop My Server. You can click on it.
How do I know it's stopped? Now I can say start my server.
Start your server, click on the small server on demand, start it up. So same thing for all of you. If you guys run into this now or when you're teaching this at your home institutions, kind of these are just going through the troubleshooting mechanisms of what could be wrong. Yeah, restarting the entire server usually works. Final last words, Lauren.
We'll see if the files are there.
know.
They're gone. Okay, it looks like the files are there.
Okay, so you want to double click the RNAseq FastQt accounts, JN. It's a little bit further.
Mm-hmm.
One more right there. Yeah.
That's 21 hours ago. Interesting. Okay.
So
So I
yeah.
have to run everything?
No. So your directory.
Well, no, it's there. It shouldn't
So your
because
directory is still there. So you're just going to have to rerun your alias or your variable one. So the one where we set up all the variables to all the paths.
down this one.
This one. Mm-hmm. Oh, it's not connected yet.
It's connecting. Yeah, I think I have a connectivity issue from Portugal tonight. I'm noticing that. So I think it may just be this happens periodically here, and it just could be that. Anyway, continue. I'll just follow along because this could take a while to connect again.
So if that doesn't connect quickly, if you
Now
want
it's idle, so now it's connected.
excellent. So go, yeah. So in it
Yeah,
already
sometimes the connectivity drops down to 3 from 40,
gotcha. Yeah.
and so it gets sluggish. Okay, well let me run this again.
So
See, now it says 1.
it won, so it ran.
Now it did 1, which it wasn't doing before. Okay, so now I can run this one.
You don't have to because those are all populated already, but it's okay if you do.
The copy there. Okay, they're there.
And then scroll all the way down to the iframe commands. So that's just, yeah, that one. Go ahead and run it. And then run the next one.
Ta-da! That was
Wonderful.
it.
Excellent.
Reboot, restart,
It's the famous,
ticket.
this is IT people get paid so much money and you call them and what they do, turn it off and turn it back on again. Okay.
So I did electrophysiology. It was the same. Is it plugged in? Is it turned on? Is the fuse okay? Slap it. And that solved 95% of the problems.
Usually, yeah. So when all else fails, turn it off, turn it back on again. Okay, great.
Thank you.
No problem. Yeah, so if any of you guys have that issue, that's kind of the troubleshooting steps that we usually go through, and ultimately turning it off, turning it back on again, hopefully will resolve most issues. Okay, now we should all be seeing this HTML report. Again, shoot it in the chat if you guys aren't seeing this. And this is interactive, so you can scroll up and down here. And this is all of the plots that we went over in the lecture. And remember, you can scroll left to right too if you're not seeing everything in your frame. And so I'm going to have you guys answer questions about this file in a minute. And so you guys remember the per-base quality score. I just went over the per tile sequence quality. Notice I didn't put that in the lecture because I'm not entirely sure how to interpret this. It has to do with the location of the clusters on the flow cell. Per sequence quality scores, again, we went over this in the lecture. So we could see what the quality scores look like for the total number of reads here on the left. Then we have per base sequence content. Sorry about that. Per base sequence content, again, we went over this during the lecture. So you see here the position of the read, and now we're looking at the distribution in percentage of A, T, Cs, and Gs. So similar to what I pointed out in the lecture, we see that wobble at the beginning. And then as Ty mentioned, it's due to the random hexamer priming that's supposed to be unbiased when we convert RNA to our cDNA. That hexamer priming is supposed to be unbiased, but it is not entirely unbiased. And so that's how we see some biases here at the beginning. And then it should eventually flatten out. So this is a pretty common graph that I see for this per base sequence content for RNA-seq data. Then if we scroll down a little more, again, we see this per sequence GC content that we went over. So again, theoretical distribution should be a normal distribution of percent GC based on the tissues that we're working with and the organism that we're using. And then your actual GC distribution is in red. So take a note of the structure of that. We're going to ask some questions. Per base N content, again, we went over this, so looking pretty clean here. We again see that tiny little blip there at the beginning, but not like really small point something percent of total reads have that, which is good. Sequence length distribution. This is looking a little weird. Not quite what we went over in the lecture. Make a note of that. Sequence duplication levels here, so that we went over too. Keep in mind that this is a subset of the data. It's only looking at the first 50 base pairs, only looking at the first 200,000 reads. So again, we have if it were deduplicated based on sequence identity from what it's having here, and then our actual duplication levels that we're seeing in the data in blue. This is the thing that I kept alluding to in the lecture, but didn't really go into detail. So these kind of duplication levels here, as well as some of the high GC content that we're seeing over here that's looking weird, we might get some information about what that is by taking a look at this table here. So this table is showing overrepresented sequences. And I think it's like if it makes up more than 0.1% of the reads in the sample. So if there's a sequence that makes up 0.1% or more of the total reads, FastQC will print out what that sequence is. Here, it'll tell you what the actual count number is, the percent of total reads that it is. And then it should give you a possible source, although I never really see this populated. So what you can do, so notice how a lot of these have high C's and G's. Which is interesting, right? So this could be some kind of contaminant. It could be maybe an adapter dimer. It could be a ribosomal RNA. So the nice part about this is if you have a lot of these and you might be concerned about them, you can kind of grab this and then do a blast search on like NCBI or something to see what it could possibly be. So that's pretty convenient. We then have our adapter content plot. So this is going to show you what the adapter content is according to the position in the read. And so this should look pretty similar to what we saw in the lecture. Yeah, so I do have, so that's the last thing that we see in the FastQC report. I don't have questions here yet. Okay, so I'm not going to ask you guys questions until we do the multi-QC report, but just want you to make a note of these things. Does anyone have any questions about any of the information that you guys have been scrolling through this FastQC report? I'm going to ask you some questions after we compile the information from everyone. The blast search suggests it's ribosomal RNA. Thank you, Rachel. Yeah, so Rachel did a blast search on some of these sequences and noted that it's mapping to ribosomal RNA, which means that, and this happens all the time, especially when you use ribodeplete. Poly-A is usually a little cleaner because you're doing a positive selection of poly-A tails, so you tend to not get as much ribosomal contamination. But when you're doing a ribodeplete, you will tend to have some level of ribosomal contamination. We actually do, I don't go over it here, but for all of the data, I do use a tool called HT-Stream to identify the ribosomal reads in each samples, and we do report it in our sample table here in in our repository. And so this is the repository for GLDS 104, the link that I put in the chat earlier. Put this link again. It's also linked in the Jupyter notebook. So this is a data set that we've been working with. And so I did analyze these data to see what the percent ribosomal RNA contamination is. Like in the assay, yeah. So it's actually, sorry, it's in the assay table. So if you go to the assay table, this has multiple assays. So if you click in the assay name, you want to go to transcription profiling, and it'll bring up the technology type, the RNAseq that was done. And so if you scroll in this table, actually, find it. No, here it is. So I actually evaluated the ribosomal RNA contamination in the raw data here. And so what you see, and so this table, and you could sort through this table. So if we do select and export columns here, I can kind of unselect all, and then I could just put in sample name. And where's our ribosomal RNA? Here it is, parameter value ribosomal RNA contamination, and you'll notice that it populates the table down here in our repository. And so I did calculate what the percent of ribosomal RNA is in these samples, and you'll notice that all of these samples that we're working with, so these are the same samples that we're working with right now. And you'll see here that in this ribosomal RNA evaluation that I did, all the samples have anywhere from 5 to 10%. This one even has 12% of the reads that are ribosomal RNA. So it's kind of making sense of why we're seeing these overrepresented sequences. As Rachel noted, she did a blast search in the ribosomal RNA. And these do have about 10% of ribosomal contamination. So it makes sense that we're seeing that in these samples. And that could also account for, because you notice that the sequence of the ribosomal RNA contamination we have has a lot of C's and G's. So that might tell us why we're seeing this peak here in the per sequence GC content. So this peak might correspond to the fact that we have ribosomal contamination a little bit in our samples here. So you get a lot of good information about the quality of your data. Does anyone have any other questions about any of these plots? I'm going to prompt you guys to ask some questions after we compile them. Okay. So remember when we ran that array script, the whole point of running the array script is so that way we ran the same command on all samples. And then keep in mind, each sample that we're run, we ran a FastQC analysis on both the forward and the reverse read. So this was the forward read that we just went over. Now we're going to view the reverse read FastQC report for the same sample, that flight rep 1 sample, and take a look at that. And then I'm going to ask you some questions. Oh, actually, then we're going to compile with multi-QC, and then I'm going to ask you some questions. So let's first... for some reason, we noticed you should technically only have to run this once, but when Lauren and I were testing, we noticed that sometimes the library like unloads or something and it doesn't work. So we do run this command every time before we look at an HTML report. So make sure that you're running that command again. And this time we're going to run, let's see, how many questions do we need to get right to pass? Yeah, all of them. No, it's okay. The whole point here is to learn. So it's okay. As long as you understand it by the time we're done, then we're satisfied. So once we import our library again to view HTML files, we can run this iframe command. And again, this time, instead of looking at our forward read, we're going to look at our reverse read for that same sample, just because I want you guys to see the comparison between the forward and the reverse for just one sample. So go ahead and run that, and you should see, again, very similar setup, right? Same thing, FastQC report, same tool. It's just that this time it's on the reverse read. So again, I'm going to take a look at the basic statistics. Remember how when we were talking, we said that the reverse read tends to have less quality than the forward read. So that actually is the case that we're seeing that here, as you notice. Again, this per tile sequence quality, I don't know, I mean, any other instructor or anyone on the meeting, feel free to chime in if you have an indication for how to interpret this. It's in the documents, but I never really pay much attention to it. And then we have this per sequence quality score here that we went over. So again, looking at the reverse reads, the reverse read for this one sample. Our per base sequence content. So again, look at distribution of our nucleotides across the position of all the reads in our sample based on percentage. So again, seeing that wobble, which is expected because of the hexamer priming to convert RNA into cDNA. We have our per GC content, so we're seeing a similar shape here in red as we saw with our forward reads. Our per base end content here, again, we see that tiny little blip at the beginning, but for the most part, no ends, which is great. Sequence length distribution, again, this is looking a little weird for raw data. Note of that. Our duplication levels, again, are here, so we're seeing some of those duplications, which, as Rachel pointed out, is likely indicative of some of the ribosomal RNA contamination that we have in the sample. So again, you could take a look at blast search, these overrepresented sequences, to try to sort that out. Adapter content, so again, taking a look, and don't forget you can scroll left to right on these. So we are seeing some adapters there in the reverse, just like we did in the forward. And so now, before I prompt you guys to ask questions, we just took a look at one sample, right? So this was our flight rep 1 sample. We looked at the forward read quality, and we just looked at the reverse read quality. And so we have 12 samples right now, right? Each a forward and reverse read. So 24 samples. I'm giving you an answer to a future question. 24 FASTQ files in total, right? Forward and reverse read for each sample. So it'd be kind of a pain to have to go through and look at the FASTQC report for each sample. And this is only 12 samples. So we have some studies that have like 50 samples per group. And so to look at all of those to try to get an idea of what the raw quality looks like across all samples can be time consuming. So there's this nifty tool called MultiQC that will compile all of the FASTQ data from all of the reads from all of our samples and create a nice comprehensive report that contains information from all of those samples. If you want to learn more about the MultiQC program, this MultiQC term here links out to MultiQC documentation. So again, welcomed and encouraged to read more about the MultiQC program. It is really nice. We're going to use it throughout the whole bootcamp. And so this command, MultiQC, actually runs pretty quickly. So we're going to run it right here on our servers all together. And so we're going to tell it, so this is the name of the command. This -n, again, you can find out what these flags and options mean by running MultiQC --help space --help, just like we did with FastQC earlier. However, for your convenience, all the options that we use in the commands, I wrote out the definitions of what those parameter options mean right on the notebook. So that way you don't have to keep going and looking into the help menu. It's all right there for you. And so we're telling it, so this -n is a prefix to name the output for all output files. So I want to put a prefix on my output files from the MultiQC. And I want to put the prefix of raw MultiQC so that all the output files will start with raw MultiQC. So I'll know that this is a MultiQC report for the raw data. We have this -o option, which is similar to the -o option in FastQC in that it directs the program of where to put the output files that are generated from MultiQC. And so we're going to output these right into that location in the directory structure that we set up at the beginning of this Jupyter notebook. So within our 00 raw data, in our FastQC reports, in a subdirectory for the MultiQC reports. And then we have a space here, and then we have this positional argument, which as defined here, is going to be the directory that contains all of the FastQC data from each sample. And so just to show you guys what that is, let me do that before I run this command. And so if you guys just want to, what command again do we use to list all files in a directory? Type it in chat, shout out loud. So cat is to view a file. Do you remember from the thing, can we cat a directory? Yeah, Kanisha, it's ls. Yeah, that's right. And just mark so that you know, if you're ever wondering, can you cat a directory? It tells you no, right? So it says that you can't do it because it's a directory. So yeah, what Kanisha had put originally was let's run ls and see what's in that directory. And what you see here, do you remember that we said that the outputs, all right, let's go all the way up here in the notebook, that the FastQC program is going to output 2 files for every data that it runs FastQC on. So it's going to run FastQC on the forward and reverse read. And so for the forward read, it's going to have a FastQC HTML as well as a FastQC.zip file. It's going to have those same files for the reverse read for every sample. And so that's what we're seeing when we ran our ls command in the directory that's storing the FastQC outputs from all of our samples. No problem, Mark. And so when we ls that, remember, this is just a variable to the path that is holding the FastQC outputs from all samples. And so as you can see, we have an HTML and a zip file. So this is the ones that we looked at, right? Rep1R1, we looked at that HTML file earlier. It also has an accompanied zip file containing all the data that's used to plot the plots that we see in the HTML file. Then we looked at the reverse read here, so we see that data here, and then we see that same thing for all 6 reps of our flight, and then for all 6 reps of our ground control. And so what MultiQC does is it's going to take that information, that FastQC data, and it's going to compile all of it into a nice summary report for all samples. So we can go ahead and run this code block, and it's going to run MultiQC, and we are actually running this right on our system right now. So this is what it looks like, the standard output from MultiQC. Luckily, this one's pretty quick, so we don't have to submit it to a job scheduler. We can run it right here, all of us together. Is there anyone that's still waiting for theirs to finish? Mark, you're still waiting? Okay. I'll give it a second. As you're waiting, Mark, for yours to finish, you just want to take a look at my screen. Oh, cool. Anyone else still waiting for theirs to finish? Good. Yeah, this tool is pretty quick. And so what it's doing, so in the standard output, the nice thing about this tool is this is one of the tools that tells you the version that it's using right up front in the standard out. So we know that this is version 1.11 of MultiQC. It's also nice that this tool will tell you if there's more updated version available. So we're actually not using the most up-to-date version because it says that a new version has been released. And then it will let you know which path it's searching, right? So this is the path that is held in this variable raw FastQC, right? So it's looking for any file in there. And now MultiQC is a tool that will compile data from a variety of different bioinformatics tools. So when you run it on a directory, it's going to look for files from any tool that it is aware of that it can compile information from. So it actually found a report that matches this tool called Snippy. I don't know what that is. I don't know if anybody else does. Might just be a coincidence. But then the thing that we're interested here is it identified files from FastQC. And how many did it identify? For everybody, type in the chat. Yeah, 24 reports. Does that make sense? Why or why not? Should it have found 24 reports? Yeah. Why, Ty? You can unmute your mic.
Because you have 12 samples, and then you have the forward and the reverse for each one.
Exactly. Yeah. And I see Susie wrote that same thing in chat. Yeah, exactly. So we expect there to be 24 reports. This is paired end sequencing on 12 samples. So we should expect to see 24 reports. So these are the things that I check for when I look at the MultiQC standard output to make sure that everything looks right. It then just gives you like a status that it's compressing the plot data, it's generating the report, and then it will let you know when it's complete. And so those are the things I look for when I look at the standard output from MultiQC. Did it find all of the reports for the tool that I was interested in? Did I see any errors? So we don't see any errors reported here from the tool, so that means that we're good. And then again, we went over these parameter definitions before. And so just I have listed here the input data that it is expecting. So it's actually for FastQC, it's looking for the data that's in that fastqc.zip file for both our forward and our reverse read for each sample. And then there's a couple of outputs that it does. So it outputs a directory that, notice how it should output with the name raw MultiQC because we gave it that in the parameter. We said, hey, all the output files, I want you to put raw MultiQC at the beginning of it. So we expect that the output data should have raw MultiQC, and then this is automatically added. So this underscore data, that's a directory containing the compiled FastQC data from all of your samples. And then we also have this HTML file, which is going to be the multi-QC report, an interactive HTML file that will show us the information from all of our samples. I do have a note here that's important when you have really, really large data sets. And so if you have more than 50 samples that are being compiled, another option that we add at GeneLab to this command is we add the option -- interactive. So multi-QC will automatically generate an interactive report if there's less than 50 samples. If there's more than 50 samples, that report will be static. You won't be able to play around with it or jump around in it unless you put this interactive flag that forces the output to be interactive. So just a heads up about that. For more information, I have a link to the document. Looks like Wygen has a question. Do you need to generate 24? Yes, good question. Yeah, absolutely. So multi-QC is expecting those fast QC reports to be there. So that's why the order that we're running these is very important, because you have to make all the fast QC reports first for all the samples. So when we ran, and I know I pre-ran all of these data, but that's why when we ran this command here, the fast QC command, we did it using this array script. So that way when we ran that fast QC command, we ran it on all 12 samples. So you have to make sure that your fast QC for all of your samples has completed before you can compile that information with multi-QC. Good question. So now we're just gonna scroll back down. I should probably hide those. Scroll back down here. And so now that we know the data that we're getting, let's take a look at the raw multi-QC report. And then I just have a note here. So if you're not sure how to interpret the information that you're seeing in the multi-QC report, multi-QC does a really good job with their documentation, especially for tools that are used frequently. So this link here actually links out to multi-QC documentation that gives information about what you should be seeing in the compilation of fast QC data in the multi-QC report, which is pretty nice. So if you want more information about what you're seeing, you click there. Okay, let's take a look at this. So first we want to run our IPython display import frame. So go ahead and run that. And then you can go ahead and run your iframe command to view the raw multi-QC report. Is there anyone who's not seeing this? Okay, great. So everyone's seeing this. So this is what it looks like, the multi-QC report of fast QC data once you compile a bunch of samples. So again, we see the same kind of similar categories here, right? We have our general statistics, we have our sequence counts, we're gonna have quality. So let's take a look at what these look like all compiled together. So now we have these general statistics, and what you see here is you have each one of your samples, and you have R1, R2, R1, R2, for each one of your samples, and it'll tell you, it kind of summarizes some of the information that it found in the fast QC reports for each one of those files. So it'll give you a summary of the percent duplications, it'll give you a summary of the percent GC content, it'll tell you the total number of sequences that it found in millions. And the nice thing about these reports is that they're interactive, right? So if I want to know, this is going to help you out answering the questions, I want to know which sample has the highest percent GC. I can click on this and it'll sort it from highest to lowest. You can click on it again, sort it from lowest to highest. You can do the same thing with any one of these categories, which is really nice. So that's the general statistics. We then have looking at sequence counts. And so this gives you a summary of the unique and duplicated reads. So a little different from the graphs with the lines that we saw. So this is how it compiles that information of unique and duplicate reads. And it's really nice too, because the other thing here is you can toggle between total number of reads, but then you could also do percentages, right? So remember, whenever you sequence data from multiple samples, it's incredibly rare, I don't know, probably impossible to get the exact same number of reads for every single sample that you sequence. And so you are going to have some variation in the total number of reads per sample. And so in order to kind of get these all on the same playing field, if you wanted to compare the raw data here, I like to look at these things in terms of percentages. So you can convert that to percentages, and now here your X category changed from originally it was number of reads in millions Or you could take a look at these and it'll give you the percent of reads for each sample. Again, blue being unique reads and black being duplicate reads. So super nice. It also has these sequence quality histograms. So you're looking at the quality here. And if you notice the qualities that are high, so if you hover over, you can see how the window changes. So you can hover over to see each sample, which is nice. When you pull these out in an HTML report, so you can actually be able to like export this plot if it's something that you wanted. You can also, I don't know if it's within the Jupyter Notebook, but in the other one, you can also toggle and show different samples. So you can select, pick and choose which samples you want to see too in the multi-QC reports, which are nice. But yeah, so you can just hover over here and see which sample is being represented. And so you notice, and this makes sense based on just looking at flight rep 1, that we noticed that the quality of our forward reads were better than the quality of our reverse reads, particularly at the end of the read. And so this is depicted here for all samples. So if you kind of toggle up and down, you'll see that this is all R1 for all the different samples, and then this is all R2 for all the different samples. Moving down to sequence quality scores. So again, this graph looks very familiar from the FastQC. So we got our Q scores down here on our x-axis, our total number of reads here on the y-axis. And again, we see now you can toggle. This is going to be all of the forward and reverse read for all of your samples. And as you move your cursor, you can see which read is what. But it gives you a nice summary of what all of them look like together. This is that tile thing. This shows this, or this is the percent of ATCs and Gs. So this is, if you guys remember the graph where we saw like the little wobble at the beginning, which is what we're seeing here. And then overall, so it does this kind of color-coded thing, and we can move up and down and see what those percent, you see that this thing over here is changing the sample name. So these are all of the samples. And these pass-fails are like cutoffs inherent to FastQC and multi-QC. I don't really pay too much attention to them. They have set things, and like Mike said, depending on the data that you're looking at and what you expect, this program might consider it a failed sample, but in reality, depending on what you're looking at, that might not be true. So I don't really pay attention to if multi-QC tells me that things pass or fail, or FastQC tells me that. So you really want to see that in the majority of the samples, you're seeing roughly equal amounts of ATCs and Gs. And then again, the nature of the RNA-seq data, because of that random hexamer priming that we have to do, we do see this wobble at the beginning. So this is where you're seeing an overrepresentation of some of the base pairs here at the beginning for all samples. Per sequence GC content. So again, looking familiar, this time we have these GC contents for all samples, and you can kind of scroll, hover over these lines and see the different samples for each one. You see pretty much consistent patterns for all samples here. And that peak here, as we know from looking at just the first sample anyways, or if you want to go to the repository and take a look, we know that we have some ribosomal contamination, which is probably the most likely cause of most of that percent GC. Per base N content, again, here you can hover over. We do notice that some of these have a little bit higher Ns here, the reverse reads specifically for some of these samples have a little bit higher percentage, and again, still about 1.5% max, so not too much. We do see some other samples with maybe a couple N's here and some of the reads. Not too concerning, in my opinion. Sequence length distribution. So again, this is pretty weird. So we see all these kind of have the same thing. So we see kind of two peaks here. Talk about that a little bit after we answer some questions. Our duplication levels is here. So I know it was summarized up at the top in that blue and black, but it does also have this graph here that we saw in the individual sample FastQC HTML file, except again, you hover over and you can see all of them. And this one doesn't have the deduplication there because it would have to have done the deduplication for everything, so it doesn't do that when it compiles it. It just tells you the percent. Again, taken with a grain of salt, because this is a very subsetted part of the reads. You can't distinguish between biological and technical duplicates, so I always take the duplication level with a grain of salt. Overrepresented sequences. So here it shows you overrepresented sequences by percentages, but it doesn't actually give you the sequences themselves. So you would have to, if you saw a lot of overrepresented sequences, or if you saw a lot of overrepresented sequences for like one of these samples or more of these samples, that would be concerning, then I would go back to the FastQC of that individual sample and do a blast search on the overrepresented sequences that are there, just to make sure it's nothing too concerning. So like what Rachel did with the rep one sample, the flight rep one sample, if we saw anything that was like crazy high, we might want to take a look at what those sequences are. Adapter content, again, taking a look here, these are all samples compiled, position, percent of sequences. So we do see adapters, and you can hover over and it'll tell you the sample name. It'll also tell you what adapter it's identifying. So we see all these are Illumina universal adapters, which makes sense. Those were the adapters that were used when this was sequenced. And then this kind of gives like a summary of all of the analyses that are done down here. And so now we have a couple of questions. So I want you guys to go through and look at these and answer these questions. I'm going to ask Lisa, are you on? Are you good?
I'm on, Amanda, and Lisa told me we should be good to go.
Awesome. So Armin, if you want to post the link. So before, guys, I know we were doing the poll to ask questions in our intro notebooks. We did want a functionality to where everyone can see what answers people are putting down without stringing their names attached to it, in case people don't want their names attached to the answers. And so I am going to request that all of you guys answer. Take your time to answer it. If you feel like you're really stumped, just put a guess in there, because I'm not going to show answers until everyone's done. So go ahead, take a look, see if you can figure it out. If you're really stumped, just put a guess or put I don't know in your answer, and we'll go over it together. So we're going to ask these questions one at a time. And so the first one is, which sample was sequenced at the greatest read depth? Which sample was sequenced at the least read depth? And I gave you a hint here. And so if you guys can all go to the link that Armin put in the chat. I'm going to put it up here too. So if you go to this link and then copy and paste the code that Armin put in the chat, and you will have that question populated.
But that is...
I think that was a question that I forgot to ask. So let's go to the next slide. Yeah, sorry. I kind of went over that question without asking it. So we'll start at the second one. So which sample was sequenced at the greatest read depth and the least read depth? And you can go ahead and put your answer here and then click submit when you're done. And Armin's going to let me know when all of you guys have answered the question, and then I'll show the results from everyone. So go ahead. Which sample was sequenced at the greatest depth and which sample was sequenced at the least depth? And you could take a look at that from the information. Yeah, you could skip the first question. I mean, I guess, actually, you can answer the first question and the second question. If you guys want to do both, you could do both.
It should just show that first slide, but if you see the other question come up, then yeah, feel free to answer it.
Yeah, feel free to answer that first question. We kind of already talked about it, that's why. But we can talk about it again. Yeah, so go ahead and answer that question. So Armin, we have 10 participants, and then TAs, instructors, if you guys could answer too. So Armin, it should match the total number of participants. We have 14 participants, including me. I'm not going to answer. So you should get 13 responses. So instructors, if you guys want to respond too, once you get those 13 responses, let me know, and then I'll...
We've got 2 so far. Keep them coming.
So which sample was sequenced at the greatest depth and which sample was sequenced at the least depth? 2 for the second question, Mike. So we're going to go back to this question here. Yeah.
We've got 4 now.
help you guys out a little bit, you want to play around with your multi-QC report. If you remember in the basic statistics up at the top, you can play around and toggle. It's an interactive thing, so we can see what's the most and what's the least. If you look at those general statistics from the multi-QC report.
We've got 4 left.
Okay, instructors, make sure you're answering too. You can sort the samples, I think. I think you can toggle on the samples. How you guys doing? Need more time?
We've got 9 now. And 10. I think we've got 2 more. And Amanda, do you have the link to be able to show the results?
Yeah.
Okay, awesome.
Okay.
All right, one more.
One more. Go ahead and put an answer there. You can either take a guess, say I don't know. Give a few more seconds, and then I'll show results. Okay, I'm gonna go ahead and show results now.
All right.
So, let's see. Was someone saying something? Okay. Let's see. Ground control rep 1 most, fewest flight rep 2, ground control rep 1, flight rep 2, ground control rep 1, flight rep 2, ground control rep 1, flight rep 2, ground control rep 1 greatest, flight rep 2 least, ground control rep 1 awesome, flight rep 2. Had a ground control rep 3, flight rep 6, okay. Ground control rep 1, flight rep 2, ground control rep 1, flight rep 2, rep 1 R1, rep 2 R. Interesting that you pointed out the actual forward reverse. Ground control rep 1, flight rep 2. Okay. So just to point out, this person who did the R1 and the R2, what should the number of reads be in the forward and the reverse samples, or forward and the reversed FASTQ file? What should the total number of reads be for forward and reverse? You guys put that in chat. Same, different? So the total number of reads in our forward FASTQ file and the total number of reads in our reverse FASTQ file. Should they be the same? Should they be different? Yeah, Chief, they should be the same. Why? You can unmute your mic. Same sequence, just opposite direction. Yes, exactly. So if we remember from our sequencing here, right? Each one of these clusters is going to be sequenced. And when you do paired end sequencing, you're going to sequence this read in the forward direction, and then you're going to sequence each read in the reverse direction. So for each cluster that you have, you're going to sequence for paired end, you're going to sequence it in one direction, and then it's going to do those washes and make the copy of it. And so we're going to have, like in this case, it'd be the purple ones that are attached to the flow cell, and then we're going to sequence in the reverse direction. So it should be the same, it's just the same insert that's going to be sequenced in both directions. So we should always have the same number of reads in our forward FASTQ file as we do in our reverse FASTQ file for the same sample. So that should always be the case. That is something also that we check. So there are tools if you tell it paired end, and we're going to say a little bit more. Might have mentioned already, but if there are some tools that when you say paired end, it's expecting the same number of reads in the forward direction it does in the reverse direction, and tools will give you errors if it doesn't have that. Okay, so back to this question of which sample was sequenced at the greatest read depth and the least. It seems like the overwhelming response was ground control rep 1 had the greatest and flight rep 2 had the least. Let's see how we can find that information. So what you can do is if you scroll up in your multi-QC of your raw FASTQC report to this general statistics. So I think, I was saying chief, so I think that you can, so if you do your sample name, it should go in, I guess it doesn't. What order is that? It's not alphabetical. How weird. But anyways, if you go to your million of sequences, so this is telling you the total number of sequences. If you're unsure, notice when I hover over that column header, it says FASTQC total sequences in millions. And so if you click on this, it'll sort for you. And you could see, so when I clicked on it, right now it's sorted from highest to lowest. And so what I see here is the highest number of reads, so the most number of sequences is in our ground control rep 1. So a lot of you guys got that. And also note that R1, the forward read and the reverse read, same exact value. It should be for every single sample. You should see the same exact value in that sample's forward and the reverse reads because it should be the same number of sequences. Again, because it's one cluster that we're sequencing in one direction and then the other direction. So it should be the same. So ground control rep 1 is the right answer for the most reads. The sample with the least reads, you could just click on this again and it'll sort it in the reverse order. And so as you guys can see, the least number of reads is going to be 65.2 million reads, and that's going to be in your flight rep 2 sample. Anyone have any questions on how we found that information out? Is that clear now? Okay, good. Yeah, so it's super useful because you can like compare your different samples and see how you're doing. The other thing, remember I told you, but ultimately we're going to be comparing differential gene expression here. So the fact that we see, actually, I might pose that question. Let me hang on to that question until I take a look at my questions. Okay, I don't ask that question. So you notice here, right, that our range now is from 85.7 million reads to 65.2. So we have from 65 to 85 million reads. So we have up to a 20 million read difference among samples. Should we be concerned? Yes, no, no, yes. Yes, unmute your mic. How are you guys feeling about that range of read depth? No one has any opinions?
Are you asking if the difference is a problem or the number of reads?
If the difference is a problem, the fact that we have a range of 20 million read difference between our samples. You think that's going to be a problem? No, if it's normalized. Okay, Jen, yeah. So you really don't want to get a too crazy range here, right? You want everything like within the same ballpark. So considering that the range of reads goes from 65 million to 85 million, well, 65 million reads, if you guys remember from our standards for ribodeplete data, we try to get it between 40 and 60 million reads. So this is a good read depth for ribodeplete in general. The fact that some samples have 20 million more reads than some other samples, that could be a little bit of a problem, but 20 million reads in the context of 60 to 85, it's like roughly what, 20 to 30% difference in reads. That's maybe a little bit concerning, but most of that, we're going to normalize for read depth later on. And so we will normalize to account for those differences. But the thing that you do want to keep in mind, like sometimes we'll get data sets submitted and there's like double the amount of reads in some samples than other. So that even though you're going to normalize for read depth, just the likelihood of, if you sequence that much more deeper, the likelihood of you picking up more transcripts that are expressed is going to increase the deeper you go. And so sometimes if you sequence way deeper in one sample than another, even if you normalize to account for that, you just might have transcripts represented when you sequence deeper that would have been in your other sample that you sequenced at less depth had you have sequenced as deep. So you really want these to be as close as possible to one another, even though you're going to normalize for it. And so I do account for that when I take a look at the data to kind of see, should I, like if you see double the number of reads in some samples than other, that I'd be a little concerned with. This within 20-30%, probably not too much. Maybe some lower expressing samples might come up in the ones that are sequenced a little deeper than the other ones. But
Amanda, is that because of the amount that was loaded differed? What causes that variation? Is it the concentration of what was loaded?
So hopefully when you load your flow cell, you should be loading the same concentration of each of your samples, right? So you measure, kind of last step here I have. So you run electrophoresis, so you determine the library quantity with a qubit or qPCR. So that's how we make sure that we load the same amount of our library for each sample. But remember, if we take a look at our bioanalyzer traces for your libraries for each sample, you kind of have a range of the fragment size, the library size. Because of that, depending on how this is shifted for each sample. So do you remember the bias of the flow cell? Is it going to favor shorter or longer fragments?
Like shorter.
Yeah, the flow cell favors shorter fragments. So when you prepare your libraries, even if they look like they're the same concentration, and when you measure it with your qubit or your qPCR, depending on what the spread of your library size looks like for each sample, like if you have one library size that tends to skew on the shorter and one library size that tends to skew on the higher, that shorter library size is going to be more likely to latch onto the flow cell. And so you're going to have a higher number of reads for those samples. And so that's why we get that variation. And so what we do at GeneLab actually to account for that is we of course take the concentration with our qPCR and balance it that way. But then because that doesn't account for if you have slightly different sizes within your samples, what we do is we actually pool all of our samples together. So we combine all of our samples like we're going to run it on the NovaSeq, on our big sequencing instrument. And we actually run it on Illumina's smallest sequencer, which is an iSeq, which only gives you about 6 million reads in total. And so, depending on how many samples you'll have like a few 100,000 reads per sample. And we'll actually take a look at the number of reads per sample on our iSeq, the smaller sequencing thing, to see how it balanced out in that smaller sequencing run, which is cheaper. And then based on how many reads per sample we have in that kind of pre-run that we do, we'll rebalance our pool. So that's how we account for if there's...
Is there flow cell variation? Do you get that affecting, or are they all on one cell?
So the flow cell, you can, yeah. So you know how the flow cells have different lanes? In the iSeq, the super small one that we do, the iSeq only has one lane. So
okay, well that will reduce the variation.
Sample there. But then when you do, you're right. So then if I do sequence on the flow cell, on a NovaSeq, a flow cell with multiple lanes, there can be bias actually on the lanes. So what we like to do is we actually try to put the same library on all lanes of the flow cell to account for that. So that way, we have the same samples running on each flow cell. So if we have a bias in one lane, where it's like, for whatever reason, maybe there's some damage to that lane of the flow cell or something, we don't get as much coverage there. We have all samples run on all the lanes of the flow cell, and so that same thing is going to be affected for all samples to kind of help account for that. But yeah, that's true. But so that's what we do, and then we kind of run that initial sequencing run on the iSeq to get a small number of reads per sample just to see how it's panning out, because the bias of the fact that shorter fragments are going to latch on more easily. And so if you have a library with shorter fragments, you're going to get more reads from that sample than from a sample that might have larger fragments. So then we like take our pool and we spike it. So if there's, when we do our iSeq run, if we see that like, oh, these 3 ground control samples have like half the reads than, you know, all the other samples, then before we run it on the NovaSeq, we do a calculation to see how much more of those samples that we would have to add to our pool so that we get roughly the same number of reads per sample. Question from Jason. After trimming, do we still need at least 40 million read depth, or is it okay for less? Yeah, so really after trimming is where you want your 40 to 60 million read range. Of course, you don't know that until after you're done trimming. So that's why we have that 40 to 60. So hopefully in our raw data, we're getting more close to that 60 end of things. So that way, once we do our trimming, that will still be left within that 40 to 60 range. Good question. Does it happen a lot, like every time that you have to rebalance after the iSeq run, or is it rare? It's pretty common. Yeah, good question. Yeah, it's pretty common just because it's very difficult to account for how it's going to play out on the run. I guess we could do a more thorough assessment of the range here and try to kind of guesstimate based on how we're seeing this smear end up. But yeah, almost every time we have to do some rebalancing. So, and there are labs too. That's why if you don't know much about your data and you outsource it, not all core facilities are going to do that initial test run. So when you do your raw data FastQC analysis, that's something that you want to check for to see if they are indeed balanced. The other thing that you can do in silico is that if you do, let's say, outsource your sequencing data, you get your raw data back and you do this analysis and you notice that there's like a really big difference in the number of reads for the samples. What you can do is, you remember how we have that cat command where we can open up an entire file? So you can open up your FASTQ file and you could like pick a certain number of lines. So that way, if you know, you get your samples back and let's say you have half your samples end up having 50 million reads and half your samples have 100 million reads, you could take your samples that have 100 million reads in each, and you could just take the first 50 million reads when you do your analysis and throw those other 50 million reads away. Of course, you're throwing away data there, but now you can kind of on the in silico side account for those read differences. I hate throwing away data, but sometimes if you get something that drastically different that you're concerned about, or I would probably run it both ways. I'd run it with all data included, and then I'd run it again if I locked off and see if I get like drastically differences in my differential expression. And if I do, I would know that it's because of the greater depth on some of the samples than others. Okay, next question. Next question is, what do you notice about the quality of the raw sequence data? Are there any differences between the forward and the reverse reads? So hopefully this is gonna be quick because I know that we answer this pretty quick. Armin, is it the same thing?
Yeah, it's the same link with the same code. It should have just automatically switched to the next slide.
Okay, yeah, so you guys should all have this question populated here.
We
So
actually have an overachiever that already answered, so.
overachievers. Yeah, so if you guys can go ahead and answer that question in the Mentimeter poll.
Mark, what are you seeing? Are you seeing the old question or? Oh, there you go.
Oh, good. Yeah, so what do you notice about the quality of the raw sequence data? Are there any differences between the forward and the reverse reads? And I know we kind of went over this a little bit, but now you're like really seeing all of the samples, quality in the forward and the reverse reads in the multi-QC report.
All right, we've got 6 answers so far.
Awesome. So what do you notice about the quality of the raw sequence data? Any differences between forward and reverse reads? I guess I could scroll up here. You want the quality plots. So here's quality plot here, but this is probably the quality plot that you want to look at. And again, to know if which one's forward, which one's reverse, you just can hover over. And I named these files. So there is R1 for the forward read and R2 for the reverse read. Kind of compare and contrast the quality of the forward and the reverse reads for all samples using this plot. How are we doing, Armin?
We've got 9 answers so far. 10. Waiting on 2 more.
And instructors, make sure you're putting in your answers too. Give a few more seconds and then I'll go ahead and share the results. Okay, submit your answer now. Let's go.
Looks like we're on 10 still, but
Okay, in the interest of time, I'm gonna show the results from everyone. Okay, so we got a decrease towards the 3' end of the reads. R2 are worse, forward better than reverse, overall good, reverse is slightly lower quality than forward better than reverse. Quality appears to be on the lower end when in regards to the reverse reads, but generally higher quality with forward reads. Reverse is worse after around the 95 mark. Forward and reverse reads should be exactly the same length, just in the opposite direction. That's true. Lower quality scores in the reverse reads towards the end, slightly higher duplication percentage in the reverse reads. Interesting. Overall, good, lots of seq duplications. R2 reads have fewer red flags than R1. Quality of the forward reads is better than the reverse reads. They're still both fairly good. Reverse is way into that. Yeah, so you guys all pretty much got this. So you notice here, looking at the mean quality scores, that the Phred score here, so the higher that Q score, the better the quality. And so what we notice is we hover that the quality between forward and reverse is pretty much the same up until, as someone pointed out, around the 95 base pair mark, we start to see this quality kind of fall off here. But it's still, you know, in this really high quality range, as you guys all pointed out. But yeah, we do see up here as we hover over, these seem to be all the forward reads from all the sample, and these seem to be the reverse reads from all the samples. Oh yeah, Armin, can you resend the website for answering the questions and the code in the chat?
yes.
So it seems like you guys all got this, so I'm not going to harp too much more on it. But yeah, so we tend to see those forward reads have slightly better quality than reverse, but thankfully the quality overall for both is pretty good. We are at our break. We do have a couple more questions on the slides, so you guys can put your answers to your questions if you want to now. So I ask, were adapters detected? And then the next question is, should we trim and or filter the data before aligning to the reference genome, and why or why not? So if you feel so inclined, if you go to the voting thing, as long as you your test, write your answers. Who's for voting?
Well, Amanda, I'd have to go through each individual slide, so I can show the next one, and that'll allow them to answer that next
All right.
question, the word adapters detected.
Okay, go ahead and put up that next question where adapters detected, and you guys can fill in that answer if adapters were detected or not, and we'll go over the answer after the break.
Sounds good.